All right. Next is Parmesh from Uni of Wisconsin. All right, Parmesh, the floor is yours.
Hello, people can hear? Thank you. So today I'm talking about privacy of genome sequencing approaches. We had one paper about GWAS. It's a slightly different problem space in that same genome thing. The work was done with my student, Sindhuja, who is going to be, she was a master's student working with me, and she's going to be doing PhD in at a different university. Okay, so here's a brief outline of my talk. I won't waste too much time, just give you an overview of the problem, discuss our ideas, and talk about it. Of course, we all know that the human genome is the ultimate identifier, and it can be thought of as a sequence of A, C, G, and T's. Those are 4 nucleotides, and that's a representation of us. And knowledge of any of the sequence of our own individual genome reveals a lot about us, right? And so, but there's a lot of interest in people sequencing their genomes for ancestry, for genetic testing, and so on. And when people submit their DNA for these various applications, we want to protect their genome from revealing to various untrusted parties. And that's the problem we're talking about, right? The challenge here is that, of course, 99% of our genome are identical between any two humans, right? And so these small variations are often in the form of what are called SNPs. SNPs are single changes in the human genome, right, at specific locations. And these changes usually define what we are, right? The 99% is common, and these small changes determine our individuality. And so the challenge for the problem that we are considering is how to protect these SNPs from being detected when you send your individual DNA for sequencing to a genome, right? So, for example, one of the common one of the very well-known SNPs for the breast cancer detection is what called BRCA, right? It occurs in chromosome 17. One of the SNPs is in chromosome 17. And if somebody else knows that you have this BRCA SNP, your propensity to breast cancer is known to them, right? So when you send your DNA for sequencing, we want to make sure that the computational algorithm that does the sequencing and alignment in the cloud cannot find out whether you have this SNP or not, right? That's the problem we are considering. This is not genome-wide studies that was there presented in the paper before, where it was aggregation of a lot of population data and trying to compare control versus test cases. Here is an individual genome sending their sequence, their DNA for sequencing, and protecting their identity. Okay. So, and one possible application scenario is an individual goes to a doctor with a health issue. The doctor's office wants to look at your genome sequence to study your drug effectiveness. This is part of genetic testing. And they send it to a sequencer to sequence. And since, of course, the doctor's office does not have the ability to sequence, they often send it to an untrusted party or a computational algorithm to do an alignment. And the question is, would that cloud service which provides this alignment, can they recognize that you have the SNP and release the data to other users? And we want to prevent them from releasing to insurance companies or other untrusted places where you want to do. Okay, so the key result is that in the paper is that we are trying to provide a method to provide using what is called read alignment. That's part of the sequencing step to reconstruct your genome after you send your DNA. We want to make sure that your SNPs are not revealed. We use a two-cloud approach, and the two clouds we assume are semi-honest but non-colluding. And the basic, the other constraint is that since the doctor's office or client that is trying to do ancestry, they don't have any computational power. They do most of the computation in the cloud, right? So this asymmetric computational environment where the privacy needs to be guaranteed, right? So the specific problem we are talking about is called read alignment. So what happens in a generic DNA sequencing situation is that your DNA is split into using what is called sonification into small segments. Your DNA is 3 billion long. It's usually split up into small sequences, maybe hundreds to thousands of base pairs long. And those 100 pairs are sequenced, right? And those sequenced regions, those 100 base pairs are aligned to your entire what's called a referenced genome. That's a public reference because all humans have roughly the same. So you align these small pieces to these public references, and then SNPs are defined with respect to that one, right? So your changes with respect to that one are what are in general SNPs, right? So for example, here's a small sequence in there among chromosome 17, right? And it gets aligned, and it will say, in chromosome 17 it is part of this section. So that's where what is called a read gets aligned. And when it gets aligned, if it doesn't exactly match, there may be a SNP there, and we don't want the cloud that does the alignment to know anything about your read or anything. In fact, the algorithm is such that it will not reveal anything about your individualized sequence, although the client, that is you as an individual, can decrypt the entire sequence to align. Okay. This is not the first, we are not the first people to look at this problem. There are a variety of solutions, two basic approaches, what I call hash table based approaches and suffix prefix based approaches to do read alignment. These are not privacy based, right? There are many read alignment solutions. We want to do them in a way that is computationally efficient, where our approach is based on one of these approaches called BWT, Burrows-Wheeler transform. It is privacy for that one. And there are other privacy-based things for other algorithms in there. I do not have much time to explain you what Burrows-Wheeler transform does, but it's a very popular real alignment technique. It is very fast, very efficient, and it does exact alignment when it is there, and when there is SNPs, it does what is called inexact alignment. It is used in many different genomic computation algorithms, right? So very popular genomic computation algorithms. What we want to do is make Burrows-Wheeler transform privacy aware. That's what we do. So it can deal with both. Of course, in real world, you have to deal with SIPs, which does inexact alignment. But I'm going to, for simplicity, going to show you with exact alignment. It exactly works for the same inexact alignment, right? So basically, if you have a sequence, what it will do is it will take the sequence, and you have this reference genome, and it will take your sequence and sort of, if it is 100 base pair long, your sequence is 100 base pair long. It will take from the last one, the rightmost thing, and sort of try to see where the substrings match in your reference genome, right? And it uses an efficient data structure called the Burrows-Wheeler transform to do that matching exactly. Okay, so right, and once it tries to match, if there are one mismatches, it can still kind of work through them, through those kinds of things. Okay, typically the Burrows-Wheeler transform is run in a cloud because although it is efficient, still it is the computational resources are large enough that you cannot do it on your phone or any individual desktop computer. It's often done in a cloud. So what you have to do is you have to send your small reads to the cloud, and that's where the privacy comes in. Right? So our approach has three key ideas. There are two components: your data structure for the Burrows-Wheeler transform. You have to obfuscate it in some way, right? And so we transform the reference genome, which is encoded in the Burrows-Wheeler transform. So we have a technique to do that transformation. Individuals' reads needs to be protected. They need to be transformed and before sending to the cloud. And three, you need to deal with inexact alignments because the goal is to identify SNPs. The individual wants to know their SNPs. So the goal, you have to make sure that it works with the inexact alignment. It's easier to work with exact alignments. There are things that you need to do to make sure it works with inexact alignments. I do not have much time to explain these 3 ideas in great detail. For you, you'll have to read the paper. But basically, let me show you what we sort of do is to transform the reads and so on. We split the BWT data structure, which is typically in one single cloud. We split that into 2 separate clouds. So part of the information is on what called cloud F, and the other part of the information is cloud L. So each cloud has only sub part of the information so that it cannot reconstruct the information from each other. So these 2 clouds have to communicate. Normally, BWT runs very fast. Our computational complexity for the computational part is exactly the same, but some penalty is added because the 2 clouds have to communicate with each other. That is not there in traditional BWT because all the algorithm runs in a single cloud. Right? Reads, which are typically an individual's genome is split into hundreds of pieces. Pieces 100 pieces long, 3 billion pieces, maybe if you have the whole genome, or if it's 1 chromosome, some still quite a lot. What we do is to hide that, we manipulate the reads and we create what I call super reads. From these super reads, it is these super reads that are sent to the cloud. Now the cloud, knowing the super read, cannot extract your individual reads. If it can extract the individual reads, it can run it on a separate BWT and recover the information. So the transformation reads occurs and it is sent to one cloud, and another data structure is sent to the other cloud to make sure when the clouds communicate, the BWT can run properly. Right? And you'll have to read the paper to see exactly what these references are. So basically, and then there are some additional things that you have to do. So you have to do some polynomial transformation and so on. And what we show in the paper is that it is correct. That is, it produces the same results that traditional BWT provides. It provides the privacy guarantees as long as the clouds are non-colluding. And the performance approach is that the penalty that is added is just communication. Right? I mean, it can be substantial. But BWT scales linearly with size of genome and so on. We show that it is exactly the same set of properties. The other thing that I did not mention is the algorithm is very highly parallel, completely parallelized. The algorithm is very highly parallelized. So the communication latency can be actually fully hidden through parallelism. Right? And with that, I want to just summarize saying that we have an approach to do read alignment in a 2-cloud system in a completely that completely protecting your SNPs so that individuals' privacy can be protected for a wide range of applications, genetic testing, ancestry, and so on. I'll stop there and answer other questions.
Thank you so much, Parmesh. All right. Any questions from the room? Online?
So I think I saw on your results slide that you mentioned you get all the matches correct, except there's one case where you don't if the first position is missed or something. But my question is, in the case of this problem of sequencing genomes and potentially delivering bad news to someone about them having some disease, or is it even okay to make one mistake? How do we handle those? Yeah.
Yeah. So basically, what just happens is it won't make any mistakes. What we show is that there is. I agree we shouldn't make any mistakes, right? But all the genome algorithms, even in BWT, are somewhat inexact. They never produce 100% results. There are always, so it's only, although I agree with that thing, that's inherent with all of these things. And there are lots of errors introduced in the whole system. Read sequencing errors. Compared to that, this error is very, very small. And when we talk about basically it's just an almost artifact. A sequence 100 long, if the last bit is incorrect, there is absolutely, you can just truncate it and get a 99. And there will be a lot of redundancy, so it will actually recover most of them.
All right. Thank you, Parmesh. I don't think we have time for an additional question, so let's thank Parmesh again, please.
Thank you.